I'm now handing the floor over to Diederick Sprangers. He is a biochemist and has been scientific coordinator of the European Network of Scientists for Social and Environmental Responsibility, ENSSER, since 2014. ENSSER supports and protects independent scientific research for the protection of the environment, biodiversity, and human health. His presentation is called Products of Genome Editing Techniques Should Be Strictly Regulated as GMOs.
Right.
Diederick, if you want to share your presentation with us, you are welcome to do so.
Right. Thank you very much, Lanka. I'll share it right away. There we go. Right. As Lanka said, I'll try and tell you a bit about the science of genome editing. Lanka has introduced me, so I won't add very much about that. And I think I should only apologize if you hear a noise in the background, because I'm working on a very old laptop and it switches on a rather loud ventilator when it gets warm. So I hope that doesn't bother you. ENSSER is an association, a European Association of Scientists, and it was actually founded in Germany. So we're an association under German law. Now, there we go. What I'm telling you is all contained in 2 publications of ENSSER, and if you want to read all the details and find all the scientific references, please take a look at these 2 publications. I was told that Christina would put the exact links in the chat, and that will make it easier for you to find them. It's a statement from 4 years ago and a very recent one, which came out just before the European Commission's report on genome editing. For some reason, yes, there we go. Okay, what we're talking about, despite claims from some people that genome editing is not genetic modification, I must stress that it is. What we're talking about is a new kind of genetic modification techniques that came about since, that was invented since about 2000. It's a variety of techniques. CRISPR is the best known one of them. But they are genetic modification, just like the older techniques of genetic modification were invented and used, have been invented and used since the 1980s. What's the difference? Well, basically, the older techniques inserted a new piece of DNA in the DNA of an organism at a random site in the genome. The genome is the whole of the DNA of an organism. And that new DNA that was introduced could be either a piece of DNA from a different organism, or it could be a piece of DNA from a very much related organism from the same species or a different variety within the species. And that gave rise to the 2 much used names that you see there, Transgenesis and Cisgenesis. When it's from a different species, foreign DNA, it's called Transgenesis. When it's from the same species, it's called Cisgenesis. Among different varieties of one species, you have different genomes, of course. So introducing a gene from one variety into the DNA of another variety may also be useful, in inverted commas. The newer techniques, most of them are called genome editing or gene editing. The main difference is that they change the DNA at a chosen site. So they're very much more directed than the older techniques. That was one of the main problems of the older techniques. They changed the DNA at a random site. It was a shot of hail really into the genome. And the newer techniques can manipulate the DNA at the chosen site, or they claim they can. And they can change the DNA there, they can delete a piece of DNA, they can insert a new piece of DNA, but they hope to do so at one particular site only. I put a question mark behind that, but at least they make a better attempt than the older techniques. That's the main difference. The older techniques, before 2000, on average, were claimed to be more accurate than conventional plant breeding when they were introduced in the 1980s. We got genetically engineered soy, we got genetically engineered maize, and a few other species since then, not very many. Cotton, rapeseed, that was about it in any big numbers on the market. They were introduced because industry said, we can change them, we can breed new varieties faster and more accurately than you can do with conventional plant breeding, just by crossing varieties. So it was said to be more accurate, and it would speed up plant breeding. It would also raise the yields of the crops. It would produce more from the crop. And thereby it would reduce hunger. These were the major claims that were made for genetic engineering. What resulted, I'm still talking about the older techniques, not about genome editing yet. In any reasonable numbers on the market, it was only 4 or 5, it has still been only 4 or 5 species. And actually only 2 varieties, only 2 traits were really changed in any markets, in any non-negligible market size. We got herbicide tolerant crops and we got insect resistant crops. There were a lot, not a lot, there were a few other traits that have been changed, and some of them have also been put on the market, but they never reached any considerable numbers on the market, a considerable portion of the market. These are really the 2 only ones of any significance. The first, very briefly, the first one, herbicide tolerance, is crops that can grow in the presence of a herbicide. A herbicide kills all the weeds that you have on your field, but most herbicides are non-selective, so they will also kill your crop unless you make your crop resistant to them. That's what this genetic modification did. And the other one, insect resistance, introduced a gene for an insecticide into the plant, so that the plant would produce the insecticide by itself, and the farmer wouldn't need to spray it on the plant. So both circled around pesticides. And the result of that was, as was predicted really, that we got superweeds, because the weeds see the enormously increased amount of herbicide that was sprayed on the crops. Only the one herbicide that the crop was resistant to was then used, and in enormous amounts. So the weeds got used to that. That's how nature works, and became resistant, and they cannot be fought. In many places in the United States, particularly, weeds are so ubiquitous and so strong against these herbicides that it's very hard to combat them. And many pieces of land have become totally unusable because of this. Similarly, with the insect-resistant crops, we got resistant insects who got used to the insecticide from the plant because it gets into the environment in much larger quantities than when the farmer would only spray it. He would spray in small amounts without genetic modification, and he would only spray when an insect plague would come about. The insect-resistant crops, the genetically modified crops, are used. They produce the insecticide during the whole season and from all the parts of the plant, so it gets into the environment in much larger quantities. So the insects get resistant to this. It's a continuation of the old pesticide war. We knew this when these GMOs were introduced. People warned for this, but it still happened. Yields were not really raised That was the biggest promise. These two crops, these two traits of herbicide tolerance and insect resistance, increased the yields a little bit, but only temporarily. There's never been an intrinsic yield rise realized by genetic modification. That is much more difficult. So only indirect small yield rises that disappeared after a few years because we've got superweeds and resistant insects. Hunger has certainly not been reduced by these techniques. The claims for the new genetic modification techniques are the following. They're said to be more accurate than the old genetic engineering techniques and therefore safe or safer. You'll notice that this suggests that the old GM techniques were not safe. That's an admission to the critics. They were said to speed up plant breeding. They would raise, it's said that it's claimed that they will raise crop yields and that they will reduce hunger. You hear all this again. You'll notice that it's exactly the same claims as with the old GM techniques. And what are the results we've seen so far? Because the new genome editing techniques have existed for over 10 years. It varies from one technique to another, but we've had them for at least 10 years now, some for longer. But so far, the only thing that has been realized with genome editing is one herbicide-tolerant crop, a variety of rapeseed, and nothing else as yet. But what do we see with genome editing? We see that it's not so accurate as people claim it to be. We see that the DNA is not only modified in the location, at the place where it's supposed to do something, we also see many off-target modifications. At other places in the genome, the DNA is changed. That was an unexpected result. So genome editing is not as accurate as people wish it to be. And this also means that genome editing is not really an adequate term. That's why ENSSER often puts it in inverted commas. The term, of course, comes from text editing. You'll know what text editing is. You change letters in a text, you change words, you change the meaning of the text thereby, but you know what you're doing. With genome editing, you don't know what you're doing because of all these mistakes. This is not the only kind of mistake. The second kind of mistake is that even if you change your DNA at the site you want, the on-target modification, then you may have another effect, a second, a third effect, more effects that you did not want than you, more than your desired effect. That happens a lot because there is hardly a single gene that has only one function. Most genes are involved in many different features of an organism. So if you change them, you don't just change one trait, one feature of an organism, you change many features at the same time. It's all a big network, really, the genes of an organism. It's a big network interconnected to everything that happens in a living organism and around it in its surroundings. Another point is that...
Sorry, Diederick, yes. Just to warn you, you have 5 more minutes.
Okay, I'm nearly done. Thanks.
Thank you very much.
One of the best known techniques of genome editing, CRISPR, is very cheap and very easy to use. You can do it in your kitchen, and people do. This happens already. Of course, people don't know enough about what they're doing then, and this allows a lot of inadvertent misuse. They use it wrongly. It allows abuse, purposeful abuse of the technique, and it allows what we call dual use. It means that military applications are also possible. The American military, the US military, is one of the big funders of genome editing research, genome editing development. Dual use means civilian and military use. The military will never say what military purpose they have in doing a certain project. They will say it has a civilian purpose. but it has a military possibility for application as well. And finally, gene drives become feasible. That is modifying not just one copy, one individual organism, but a whole population. For instance, a whole population of an exotic species that have been introduced in a place where it shouldn't live by people. And by modifying the whole population at the same time, people want to kill them all and get rid of an exotic species. Of course, this is very dangerous because you're manipulating evolution, really. And this is another level of risks. So how come that genome editing and transgenesis, all the GM techniques fail to deliver on their claims? Well, that's basically because all these traits that they want to change, the yield, the intrinsic yield, drought tolerance, disease resistance, etc., each one of these traits requires multiple genes. And many scientists even now say that each single trait requires a big portion of the whole genome, maybe the whole genome altogether. It is simply technically unfeasible to modify so many genes at once in a controlled manner and free of undesired effects. So for short, achieving a desired effect of genetic modification is uncertain. But the one thing that is certain is that you will get undesired effects. So the efficacy of GM and therefore the safety are at risk. And this is not something new. We've known this for decades. One gene, one trait relationships are very rare in life. DNA is not the blueprint of life. It's more complex. And also the root causes of hunger are not just related to crop yield. They're more related to conflicts, to poverty, to exclusion, etc., which means that by raising yields, you will not beat hunger. There is, in fact, already much more food on the world than is required for feeding the world's population. So there's no need to raise yields. So my conclusions are, firstly, that GM agriculture cannot and will not solve hunger because science shows that it is unfeasible and because everyone can be fed with the world's available food. And genome editing must be strictly regulated because it is inherently unpredictable, just like transgenesis and cisgenesis, and because it allows more abuse, more inadvertent misuse and dual use than transgenesis did, and also because it allows developing gene drives. So yes, the EU GMO legislation must be modernized. Extra rules should be included to control these new risks, and the rules that are currently in place must certainly be maintained. That's all. Thank you.
Thank you so much, Diederick. I think this was very important for a first launch of this topic in Portugal, and so that people are a little bit less afraid to start researching more and start talking about it, because it's in fact not so mystical as the industry would like us to believe. They're actually quite, there's actually a quite clear way. It doesn't even have to be a very scientific way to explain what these new genetic techniques are, where they come from, what they are planning to do.